Hi everyone, let's start today's session. I'll share my screen. Okay, can someone unmute and let me know if my screen is visible and if I'm audible? Anyone? Okay, great. Thank you. So, good evening, guys. Today is the third live session. Basically on this course Biochemistry, that is the course instructor is Professor K. Subramaniam from IIT Madras, and in this course basically we are going to discuss some of the assignment questions from previous year, and we'll also discuss the concepts behind those questions so it's easier for us to solve similar type of other questions. Okay, so we'll move to the first question for today, and we'll discuss the options, we'll discuss the concepts, and then we'll jump on to the solution. Okay, so the first question for today is this: which of these is not a fat soluble vitamin? Anybody? Which one of these is not fat soluble? So there are fat soluble vitamins and there are water soluble vitamins, right? So which of these is not a fat soluble vitamin? Anybody? Anybody? Okay, one second. Yeah, so someone said C. Does anybody have any different answer? Yeah, it's C. C is not a fat soluble vitamin. So the C is basically a water soluble vitamins, and the vitamin B complex. There are also water soluble vitamins like thiamine, riboflavin, niacin, pantothenic acid, pyridoxine, biotin, folate, and cobalamin. All these are water soluble vitamins. So the four fat soluble vitamins are vitamins A, D, E, and K. So these vitamins are absorbed more easily by the body. Okay, in the presence of dietary fat. The water soluble vitamins are not stored in the body, and the 9 water soluble vitamins are the vitamin C and all the 8 B vitamins. Okay, and any leftover or any excess amounts of these water soluble vitamins, they basically leave the body through the urine. So they have to be consumed on a regular basis to prevent shortages or deficiencies in the body. Okay, and the exception to this is vitamin B12, which can be stored in the liver for many years. And each of the vitamins have important jobs in the body. Okay, if any vitamin deficiency occurs when you do not get enough of a certain vitamin. Okay, and they obviously can cause a lot of health problems. So different vitamins and help in different functions, and the deficiency of different vitamins will lead to different deficiency, like different disorders or different problems, health problems. So we'll discuss that. So what are vitamins? Vitamins are essential organic nutrients required in small amounts. So they cannot be synthesized by the body, must be obtained by outside sources like diet, rumen, bacteria, and the sun. And they are required for growth, maintenance, reproduction, and lactation. Okay, so there are 13 essential vitamins. This means that these vitamins are required for the body to work properly. The vitamin A, C, D, E, K, and B1, vitamin B1, which is thiamine, vitamin B2, which is riboflavin, vitamin B3, which is niacin, vitamin B6, which is pyridoxine, vitamin B12, which is cyanocobalamin, vitamin B5, which is pantothenic acid, vitamin B7, biotin, and vitamin B9, folic acid. Okay, so these are the essential vitamins. So what are the fat soluble vitamins? So these are the 4 fat soluble vitamins: A, D, E, and K. Okay, so fat soluble vitamins can be stored in the body's liver, fatty tissue, and muscles. And the 4 fat soluble vitamins are these, and these vitamins are absorbed more easily by the body in the presence of dietary fat. Okay, what are water soluble vitamins? They are not stored in tissues, and thus they should have a constant supply. The examples are all the B vitamins and vitamin C. So what is the function, deficiency, what are the signs and sources of vitamin A? So vitamin A, the function is it helps in developing healthy skin and nerve tissue. It also helps in building up resistance to infection. It functions in eyesight and bone formation. And all animals require a source of vitamin A, and it is very important in case of pregnant females. So vitamin A basically helps form and maintain healthy teeth, bones, and soft tissues, mucous membranes, and skin. So what are the deficiency signs? So there will be retarded growth in the youngsters. There will be development of a peculiar condition around the eyes known as xerophthalmia. There can be night blindness and reproductive disorders. So these are the deficiency signs. What are the sources? Whole milk, carotene, animal body oils like cod, fish and tuna, legume forages and all these are the sources, and it can also be synthetically produced. For vitamin E, the function will be normal reproduction. It is basically an antioxidant, also known as tocopherol. It helps the body to form red blood cells and also use vitamin K. So deficiency signs can be poor growth, okay, muscular dystrophy, white muscle disease in ruminants and swine, and stiff lamb disease. It can basically affect the nerves and vessels, okay. The sources can be synthetic for poultry and swine, cereal grains, wheat, germ oil, green forages, protein concentrates, oil seeds, etc. So vitamin E rapidly destroys in rancid or spoiled fats. That is why these may cause white muscle disease. So utilization of vitamin E is apparently dependent on adequate selenium in the system. Okay. So vitamin E, as I said, it's an antioxidant, also known as tocopherol. Okay. What is vitamin D? So vitamin D is also known as the sunshine vitamin, since it is made by the body after being in the sun. So 10 to 15 minutes of sunshine 3 times a week is almost enough apparently to produce the body's requirement of vitamin D for most people at most latitudes. Now people who do not live in sunny places may not take enough vitamin D. It is very hard to get enough vitamin D from food sources alone. So vitamin D helps the body absorb calcium. You need calcium for the normal development and maintenance of healthy teeth and bones. And it also helps maintain proper blood levels of calcium and phosphorus basically. So its function is essential for the proper utilization of calcium and phosphorus to produce normal healthy bones. And the deficiency signs can be retarded growth, mishappened bones, rickets basically, and osteoporosis. Sources can be whole milk, forage crops, fish liver oils, irradiated yeast. All of these are sources of vitamin D. Coming to vitamin K, it is necessary for the maintenance of normal blood coagulation. Okay, because without it, blood would not coagulate normally. And some studies suggest that it is important for bone health as well. So what are the deficiency signs? Blood loses its power to clot, or the time needed for clotting is longer, and serious hemorrhages can result from slight wounds or bruises. What can be the sources of vitamin K? Green leafy forages, fish meal, liver, soybeans, rumen and intestinal synthesis, and synthetic compounds. All these can be sources of vitamin K. Coming to vitamin C, which is none other than ascorbic acid. It is basically an antioxidant that promotes healthy teeth and gums. And it helps the body absorb iron and maintain healthy tissue. It is also essential for wound healing. So it doesn't affect on the metabolism of calcium in the body. It's not required in usually farm animals. And the deficiency signs are like human deficiency can be scurvy, swollen and painful joints and bleeding gums, and brittleness of bones. Okay. And sources obviously any kind of citrus fruits, tomatoes, leafy vegetables and potatoes. These are sources of vitamin C. Coming to vitamin B1, which is none other than thiamine. So it helps the body cells basically change carbohydrates into energy. So getting enough carbohydrates is very important during even pregnancy and breastfeeding. So and it is also essential for heart function and healthy nerve cells. So it's required for the normal metabolism of carbohydrates, and the deficiency can be loss of appetite, muscular weakness, severe nervous disorders, general weakness and wasting. Sources can be raw whole grains, and especially their seed coats and embryos, fresh green forage and yeast, milk and rumen synthesis. Coming to vitamin B12, riboflavin, it basically works with the other B vitamins. It is important for body growth and the production of RBCs or red blood cells. It is necessary for the normal embryo development, important in the metabolism of amino acids and carbohydrates. So deficiency of vitamin B2 will cause poor reproduction characterized by small litters and deformed young, curly toe paralysis in chicks, digestive disturbances, general weakness, and eye abnormalities. The sources can be milk and dairy byproducts, yeast, green forages, whole grains, wheat bran, and synthetic riboflavin human synthesis. Okay. So, what are some of the mineral facts? Facts: essential inorganic nutrients required in small amounts, as many as 20 minerals may be required. These are required for growth, maintenance, reproduction, and even lactation. Okay. So, what about calcium? Calcium, we know that the major component of bones and teeth and essential in blood coagulation, nerve and muscle function, and milk and egg production. And the deficiency of calcium may result in retarded growth, deformed bones in young animals, osteoporosis in older animals. And the sources for calcium can be milk, limestone, oyster shells. Sodium chloride again is considered together because of a close biochemical relationship and are provided as common salt. Okay, sodium and chloride. So their function is required for the formation and retention, concentration and pH of body fluids such as protoplasm and blood. And it's also important in the formation of digestive juices and functions in nerve and muscle activity. And the deficiency can result in poor condition and depressed appetite. Okay, and the sources can be salt supplements and injectable products. Next, phosphorus is essential for the formation of bones, teeth, and body fluids. It's required for metabolism, cell respiration, and normal reproduction. Deficiency can be similar to calcium deficiency, that is lack of appetite, poor reproduction, and unrich appearance. And the sources can be dicalcium phosphate, bone meal, and low fluorine phosphates. Next, we come to potassium or K. Its function is retention and formation of body fluids, pH, concentration of body fluid, and human digestion. And the deficiency signs can be non-specific and unlikely under most conditions, but may have decreased feed consumption and efficiency. And sources in roughages, rains are less than roughages. Then manganese, it helps in fetal development, adult development, milk production, and skeletal development. Deficiency can lead to abortions, reduced fertility, deformed young, and poor growth. And sources can be most used like trace mineralized salt. For copper, copper should be present in animal tissues for iron to be properly utilized, hemoglobin formation, and synthesis of keratin for fur and wool growth. The deficiency can lead to poor pigmentation of feathers, stringy wool, swayback lambs, lack of muscle coordination, and anemia. Sources can be any kind of copper salts and forages. Then we come to iron, which is essential for the function of every organ and tissue of the body, because obviously we know the importance of hemoglobin. Deficiency can lead to seldom occurs in old, like it can be seldom in old animals. It can lead to nutritional anemia, labored breathing, and pale eyelids, ears, and nose. And sources can be copper or trace mineralized salts. Okay. Next, we come to cobalt. Obviously, it's required as a nutrient for the microorganisms in ruminants and thereby helps in the human synthesis of vitamin B12. Okay, cyanocobalamin. Basically, and because swine cannot manufacture B12 from cobalt, the diet is supplemented with vitamin B12 instead. Deficiency can lead to lack of appetite, loss of weight, rough hair coat, anemia, decreased milk and wool production, and death even in some cases. Sources can be legume forages and salt containing cobalt. Magnesium, similar to calcium and phosphorus. Deficiency signs can be irregular heartbeat and severe kidney damage. Sources are obviously mineral supplements and ordinary feeds. Okay, so we have talked about nutrients, which is basically chemical substances in food that are used by the body to produce energy and tissues. We have talked about vitamins, which are essential organic nutrients required in small amounts that cannot be synthesized by the body, and they are required for growth, maintenance, reproduction, and lactation. So decline in health due to the lack of a vitamin ratio is known as vitamin deficiency. Okay, we can talk about vitamins B6 also. Vitamin B6 is pyridoxine, which helps form red blood cells and maintain brain function. So vitamin B6 plays an important role in the proteins that are part of many chemical reactions in the body. So the more proteins we eat, the more pyridoxine your body requires. Okay. Then vitamin B12 or cyanocobalamin is important for metabolism. It also helps form red blood cells and maintain the central and peripheral nervous systems. Vitamin C, D, we've already talked about. We have talked about E and K. Let's talk about biotin. Okay, biotin is vitamin B7. So what happens? It's essential for the metabolism of proteins and carbohydrates and in the production of hormones and cholesterol. Then let's talk about niacin, which is vitamin B3. Niacin is a B vitamin that helps maintain healthy skin and nerves. It also has triglyceride lowering effects at very higher doses. Okay, then we come to folate or folic acid or vitamin B9, which works with vitamin B12 to help form RBCs. It is needed for the production of DNA, which controls tissue growth and cell function. And any like low levels of folate are linked to birth defects such as spina bifida. And many foods are now fortified with folate in the form of folic acid. If we come to pantothenic acid, it is vitamin B5. It's essential for the metabolism of food. It also plays a role in the production of hormones and cholesterol. Then vitamin B2, which is riboflavin. We have already talked. It's important for body growth and production of RBCs. Okay. And thiamine, which is vitamin B1, helps the body cells change carbohydrates into energy. Okay. So these are different vitamins. So we have talked about fat soluble vitamins, which can be stored and accumulated in the liver and other fatty tissues. We have talked about water soluble vitamins, which cannot be stored in the tissues and must be provided regularly as deficiencies can develop in a short time. And minerals, which are essential inorganic compounds needed in small amount, required for growth, maintenance, reproduction, and lactation. Whereas macrominerals are required in large amounts, microminerals are required in small amounts. Okay, so the answer to the previous question was C is not a fat soluble vitamin, it's a water soluble vitamin. A, D, E, and K are fat soluble vitamins. Coming to the next question for today. Cellular membranes are typically made of polysaccharides, nucleic acids, phospholipids, and collagen. Anybody? Any other answer from anybody else? Yeah. Anybody has any other answer? Okay, so we'll move. Okay. One second. Yeah. So the correct answer is phospholipids. Okay. So a little bit detail about the membranes. So plasma membrane is a boundary that separates a living cell from its surroundings, and basically it exhibits selective permeability. Now what do you mean by selective permeability? It allows some substances to cross it more easily than others. Okay, so the plasma membrane is not permeable to all and everything. Okay, it is selectively permeable, which means it allows certain substances to pass through. Okay, so and proteins, lipids, and carbohydrates basically are the primary components of the plasma membrane. Now, cell membranes are fluid mosaics of lipids and proteins. Phospholipids are the most abundant lipid in the plasma membrane. They are basically amphipathic molecules containing hydrophobic and hydrophilic regions. So what are amphipathic molecules basically? So all of the lipid molecules in cell membranes are amphipathic. That is, they have a hydrophilic, which is water loving or polar end, and a hydrophobic or water fearing or non-polar end. And the most abundant membrane lipids are the phospholipids. They have a polar head group and two hydrophobic hydrocarbon tails. Okay, so basically amphipathic means amphipathic molecules have parts that are hydrophobic and parts that are hydrophilic. So the two hydrocarbon chains of phospholipids are composed of chains of carbon atoms non-covalently bonded to hydrogen atoms making the tails hydrophobic. Whereas the phosphate group on the phospholipid head is charged, making it hydrophilic. Okay. So phospholipids are amphipathic molecules containing hydrophobic and hydrophilic regions. Coming to the fluid mosaic model. So the fluid mosaic model states that a membrane is a fluid structure with a mosaic of various proteins embedded in it. One second. One second. Exactly. So the fluid mosaic model was kept forward by Singer Nicholson. Okay, and basically in 1972, I guess, it was proposed by Singer and Nicholson to explain the structure of the plasma membrane. So the model has evolved somewhat over time, but it still best accounts for the structure and function of the plasma membrane as we now understand them. So the fluid mosaic model describes the structure of the plasma membrane as a mosaic of components, including phospholipids, cholesterol, proteins, and carbohydrates, that gives the membrane a fluid character. Now, plasma membranes range from 5 to 10 nanometer in thickness. Okay, so the proportions of proteins, lipids, and carbohydrates in the plasma membrane vary with cell type. Okay, and obviously there are different components and functions of the plasma membrane. The phospholipid is the main fabric of the membrane. The cholesterol is attached between the phospholipids and between the two phospholipid bilayers. The integral proteins are embedded within the phospholipid bilayers, may or may not penetrate through both the layers. The peripheral proteins on the inner or the outer surface of the phospholipid bilayer, they're not embedded within the phospholipids, and there are carbohydrates, components of glycoproteins and glycolipids, and those are generally attached to proteins of the outside membrane layer. Okay, so the main fabric of the membrane is composed of amphipathic or dual-loving phospholipid molecules. Okay, so membranes have been chemically analyzed and found to be made of proteins and lipids, and scientists studying the plasma membrane reason that it must be a phospholipid bilayer. So you see this. So the hydrophilic or the water-loving areas of these molecules are in contact with the aqueous fluid both inside and outside the cell. Okay, hydrophobic or water-fearing molecules tend to be the non-polar parts. So a phospholipid molecule consists of a 3-carbon glycerol backbone with 2 fatty acid molecules attached to carbons 1 and 2, and a phosphate containing group attached to the 3rd carbon. And this arrangement gives the overall molecule, a structure. It has a polar character or negative charge, the polar head groups, the phosphate containing, and an area called the tail which has no charge. So they interact with other non-polar molecules in chemical reactions, but generally do not interact with polar molecules. So when placed in water, hydrophobic molecules tend to form a ball or cluster. So the hydrophilic regions of the phospholipids tend to form hydrogen bonds with water and other polar molecules on both the exterior and the interior of the cell. Thus the membrane surfaces that face the interior and exterior of the cell are hydrophilic. In contrast, the middle of the membrane is hydrophobic and will not interact with water. Therefore, phospholipids form an excellent lipid bilayer cell membrane that separates fluid within the cell from fluid outside the cell. Okay. Now, proteins make up the second major component of the plasma membrane. Integral proteins are, as the name suggests, integrated completely into the membrane structure, and the hydrophobic membrane spanning regions interact with the hydrophobic region of the phospholipid bilayer. Single pass integral membrane proteins usually have a hydrophobic transmembrane segment that consists of 20 amino acids, and some span only part of the membrane associated with the single layer, while others stretch from one side of the membrane to the other and are exposed on either side. Now, there are some complex proteins that are composed of up to 12 segments of a single protein which are extensively folded and embedded in the membrane. So, this type of protein has a hydrophilic region or regions and one or several mildly hydrophobic regions. So this arrangement of regions of the protein tends to orient the protein alongside the phospholipids and with the hydrophobic region of the protein adjacent to the tails of the phospholipids and the hydrophilic region of the regions of the protein protruding from the membrane and in contact with the cytosol or the extracellular fluid. Okay. So in 1935, Hugh Dawson and James Danieli proposed a sandwich model in which the phospholipid bilayer lies between the two layers of the globular proteins. And later studies found problems with this model, particularly the placement of the membrane proteins, which have hydrophilic and hydrophobic regions. Then in 1972, Singer-Nicholson proposed that the membrane is a mosaic of proteins dispersed within the bilayer, with only the hydrophilic regions exposed to the water. So if you can see over here, these are integral membrane proteins. This might be an integral, might be peripheral also. Since it goes to the entire thing, that's this embedded in the entire bilayer, that is an integral membrane protein. Okay. So freeze fracture studies of the plasma membrane supported the fluid mosaic model. Basically, freeze fracture is a specialized preparation technique that splits a membrane along the middle of the phospholipid bilayer. Okay. So if you can see over here, this is a technique of freeze fracture. So this is the inside of the extracellular layer, where this is the inside of the cytoplasmic layer. So phospholipids in the plasma membrane can move within the bilayer. Okay, so they have diffusional properties. So most of the lipids and some proteins drift laterally. So there's rarely an event where a molecule flip-flop transversely across the membrane. Okay, so this is the structure of the bilayer. If you can see, there are obviously fibers of ECM or extracellular matrix outside. You have the glycoprotein, carbohydrates, and cholesterol, peripheral proteins. So if you see, this is an integral membrane protein. This is embedded into the entire bilayer. While these are peripheral proteins, they are not really embedded into the bilayer. And these structures are cholesterol. Okay. So lateral movement occurs apparently approximately according to 10 to the power 7 times per second, whereas flip-flopping is very, very rare. So that is quite rare. So as temperatures cool, membranes switch from a fluid state to a solid state, and the temperature at which a membrane solidifies depends on the types of lipids. So membranes rich in unsaturated fatty acids are more fluid than those rich in saturated fatty acids. So membranes must be fluid enough to work properly. They're usually about as fluid as salad oil. Okay. So the steroid cholesterol has different effects on the membrane fluidity at different temperatures. So at the physiological temperature, 37 degrees Celsius, cholesterol restrains movement of the phospholipids. And at cool temperatures, it maintains fluidity by preventing the tight packing. Okay. So you can see in the figure A, we see unsaturated versus saturated hydrocarbon tails. And in figure B, see in the unsaturated portion, see how the fluid mosaic, how the phospholipid bilayer is arranged. So there are haphazard tails moving here and there. However, in case of saturated hydrocarbon tails, it looks much more rigid and much more tight. Whereas in image B, we see that the cholesterol within the animal cell membrane, it basically makes the cell membrane more rigid. If you can see over here, it's very rigid. So that's the function of cholesterol. So what's the evolution of differences in membrane lipid composition? So variations in lipid composition of cell membranes of many species appear to be adaptations to specific environmental conditions. Very true. So we know that there are very different types of organisms living in different types of conditions. If we know about extremophiles, there are organisms or bacteria, basically microorganisms that can survive in very extreme conditions, like maybe in high thermal conditions, high temperature, or very low temperature conditions, high pressure conditions or very low pressure conditions, high in different types of pH, acidic pH conditions or alkaline pH conditions. So these organisms can survive only if they have their entire protein machinery and everything inside the body, the DNA, RNA, everything in proper condition. So the lipid membranes have evolved, okay, and the difference has evolved throughout, and the variations basically point towards the adaptations of specific of different organisms to their specific environmental conditions. That makes sense. So ability to change the lipid compositions in response to the temperature changes is evolved in organisms that live where temperatures vary. So if there are organisms that live where temperature vary throughout the year, a lot throughout the year, then the organism staying there would have the ability to change the lipid compositions in response to that effect, because evolution would have done its job. So what are membrane proteins and their function? So membrane is a collage of different proteins often grouped together embedded in the fluid matrix of the lipid bilayer, and proteins determine most of the membrane's specific functions. Now, peripheral proteins are bound to the surface of the membrane. Integral proteins are penetrate the hydrophobic core. So integral proteins that span the membrane are called transmembrane proteins. And the hydrophobic regions of an integral protein consist of one or more stretches of nonpolar amino acids, often coiled into alpha helices. Okay. So you can see this is how a protein looks like inside the lipid bilayer. You have the N terminus, you have the C terminus, and you have the alpha helix. Okay. And we can see the cytoplasmic side and the extracellular side. Now, what are the 6 major functions of membrane proteins? They help in transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and attachment to the ECM, the cytoskeleton and ECM. If you see, this is a transport protein that is helping to transfer this blue solute to this side. Okay. So if you see, it is transferring the blue solute to this side. Again, in this case, it is transferring the red solute to this side. Can anybody look at the schematic and just guess why is ATP shown in this case and not in this case? So this schematic basically means that this transport of red solute across the membrane requires ATP. But there is no ATP shown here in this case. Can someone look at the image and try to guess why? Yeah, so active transport, why? How can you look at it and say it's active transport? Like it's using ATP. But why do you say it's active? Why cannot it be passive? Correct. That's what I wanted to hear. Yes, you're correct. So if you see from the image, you can see that red is going against the concentration gradient. So basically, we know that things will flow from higher concentration to lower concentration. But if you see this, the red solute over here, the higher concentration is on the outside. Outside means this side. And lower is on this side. But if you see, it's going from the lower concentration to the higher concentration. Which is why it requires energy to drive this active transport. Okay, so that is why it needs ATP. Also, there are proteins on the phospholipid bilayer that are involved in enzymatic activity. And the most important is obviously signal transduction. So cell signaling happens from the membrane, starts from the membranes. Any kind of receptor, proteins can act as receptors. Any kind of signaling molecule can come and attach to the receptor, and there is a signaling cascade basically that starts downstream. Proteins on the membranes can also help in cell-cell recognition. As you can see here, it can help in intercellular joining. It can help in attaching to the cytocellular and extracellular matrix. Now, membrane structure results in selective permeability. So, as I said, that plasma membranes have selective permeability, which basically means that they allow certain molecules to pass through while acting as the barrier for the others. Now we know a cell must exchange materials with its surroundings, and this process is totally controlled by the plasma membrane. And plasma membrane is selectively permeable. That means they regulate the cell's molecular traffic. So hydrophobic molecules such as hydrocarbons can dissolve in the lipid bilayer and pass through the membrane rapidly. Okay. Now, polar molecules such as sugars do not cross the membrane easily. Okay, there is when the selective permeability of the lipid bilayer will come into place. So now there are certain transport proteins that allow passage of hydrophilic substances across the membrane. So some transport proteins called channel proteins, they have a hydrophilic channel, and through which certain molecules or ions that they can use as tunnel basically to pass through. And channel proteins called aquaporins basically facilitate the passage of water. It's pretty self-explanatory from the name, aquaporins facilitating the passage of water. Now there are other transport proteins called carrier proteins that bind to molecules and change shape to shuttle them across the membrane. Okay, and a transport protein is usually specific for the substance it moves. It's nothing non-specific that it will help all the substances from, it will help everything, all the movement, but transport protein is very specific for the substance it moves. Now, passive transport is diffusion of a substance across the membrane with no energy investment. Diffusion is the tendency for molecules to spread out evenly into the available space. So, although each molecule moves randomly, diffusion of a molecule, population of molecules may be directional. So, at dynamic equilibrium, as many molecules cross the membrane in one direction as in the other. Okay, so if you can see, this is diffusion of a solute. Okay, these are molecules of a dye, and there is water on the other end, and then see there will be diffusion, net diffusion basically, and at one point there will be equilibrium, almost equivalent number of molecules on both the sides. What happens when there is diffusion of two solutes? So for the two solutes also there will be net diffusion for both the solutes to either sides, and at one point there will be a time when there is almost an equilibrium type of condition where there are equivalent number of both the molecules on either sides. The substances diffuse down their concentration gradient as I was speaking. So the region along which the density of a chemical substance increases or decreases. So no work must be done to move substances down the concentration gradient. So diffusion of a substance across the biological membrane is passive transport because no energy is expended by the cell to make it happen. So when diffusion is happening down the concentration gradient, the cell does not need to spend any energy for that to happen. So that is passive transport. Okay. In exocytosis, for example, transport vesicles migrate to the membrane, fuse with it and release their contents. And many secretory cells use exocytosis to export their products. This is one way of how the cells throw away, release their contents outside. In endocytosis, the cell takes in macromolecules by forming vesicles on the plasma membrane. And basically, it's a reversal of exocytosis involving different proteins. It can be phagocytosis, which is basically cellular eating. When there is a microbe or anything pathogenic attack on the cells there will be immune cells that will phagocytose the bacteria or pathogen. That is phagocytosis, cellular eating. Then we have pinocytosis, that is cellular drinking. And then we have receptor mediated endocytosis. Okay, so basically in phagocytosis, the cell engulfs a particle in the vacuole, and the vacuole fuses with the lysosome to release the particle. So we have talked about lysosome in one of the previous classes. Lysosome is the suicidal bag of the cell. It contains a lot of hydrolytic enzymes that are active at acidic pH. Correct? So what happens when a pathogen is taken inside, and the lysosome also maintains an acidic pH. So when when a pathogen is taken inside, the immune cell will do phagocytosis. It will engulf the foreign pathogen and then will fuse with the lysosome. So now it's the job of the lysosome to basically degrade or digest the foreign pathogen using its hydrolytic enzymes. In pinocytosis, the molecules are taken up when extracellular fluid is gulped into tiny vesicles. Okay. And in receptor-mediated endocytosis, the binding of ligands to receptors triggers vesicle formation. So when there's a binding event happening, an interaction event happening where ligands are binding to receptors, and this is basically triggering the vesicle formation. So a ligand is any molecule that binds specifically to a receptor side of another molecule. So if you see, this is how phagocytosis occurs. So this is a food or other particle basically, and there will be a phagocytosis happening, and the food particle will be taken inside. For example, in case of pinocytosis, this is how pinocytosis happens. And in case of receptor-mediated endocytosis, there need to be proper interaction between the receptors and the uh, molecules outside, which will ultimately lead to the endocytosis process. And phagocytosis, it can be a bacterium. So any amoeba engulfing a bacterium via phagocytosis TEM. So if you see, this is pseudopodium of amoeba, and this is the bacterium. This is the food vacuole of the bacterium. Food vacuole basically, and you can see that the amoeba is engulfing the bacterium, and this has been recorded and monitored using TEM. So this is how it happens. Schematic to show how it happens. Then we have pinocytosis. So these are pinocytosis vesicles forming in a cell lining a small blood vessel. Okay, and these are also TEM images. If you see on the right, we have a schematic for pinocytosis. Then we move on to receptor-mediated endocytosis of the plasma membrane, and there are various coat proteins. Okay, so a coated vesicle forms during the receptor-mediated endocytosis. You can basically extrapolate this image to how this looks. So you can see this is how the receptor-mediated endocytosis occurs. So that was that. So in case of a little bit more about the phospholipid membranes, so carbohydrates has been known as the third major component of the plasma membranes. They are always found on the exterior surface of cells and are bound either to proteins or to lipids. And these carbohydrate chains may consist of 2 to 60 monosaccharide units and can be either straight or branched. And along with peripheral proteins, carbohydrates can form specialized sites on the cell surface that allow cells to recognize each other. And this recognition is very important to cells as it allows the immune system to differentiate between self cells and foreign cells. Similar types of glycoproteins and glycolipids are found on the surfaces of viruses and may change frequently, preventing immune cells from recognizing and attacking them. And these carbohydrates on the exterior surface of the cell, the carbohydrate components of both glycoproteins and glycolipids are collectively referred to as the glycocalyx. And the glycocalyx is highly hydrophilic and attracts large amounts of water to the surface of the cell. And that aids in the interaction of the cell with the water environment and in the cell's ability to obtain substances dissolved in the water. Okay. So what were the key points? What are the key points to be remembered? The main fabric of the membrane is composed of amphiphilic or dual nerving phospholipid molecules. Integral proteins are the second major component of plasma membranes and are integrated completely into the membrane structure, with the hydrophobic membrane spanning regions interacting with the hydrophobic region of the phospholipid bilayer. And carbohydrates are the third major component of plasma membranes and are always found on the exterior, surface of the cells, where they are bound either to proteins forming glycoproteins or to lipids forming glycolipids. Moving on to the next question for today. The pH of a buffer is calculated using Michaelis-Menten equation, Henderson-Hasselbalch equation, Henry's law, or Le Chatelier principle. Anybody? Yeah. Okay, can someone tell me what Michaelis-Menten equation is used for? What do we do with Michaelis-Menten equation? Anybody? So Michaelis-Menten equation is basically used in biochemistry, and it was named after Leonard Michaelis and Maud Menten. And basically it's a simplest case of enzyme kinetics, okay, applied to enzyme catalyzed reactions of one substrate and one product. So it takes the form of a differential equation describing the reaction rate V, formation of the product P, and filled by the substrate. The equation is called as the Michaelis-Menten equation, which is basically developed to see enzyme kinetics. Okay. So, but the pH of a buffer is calculated using Henderson-Hasselbalch equation. That's correct. Okay. So, the correct answer is Henderson-Hasselbalch equation. So, moving on, we'll talk a little bit about the ionization of water. So, water molecules have a very slight tendency to ionize to hydrogen ion and a hydroxide ion. Okay, H plus and OH minus. So actually free protons do not exist in solution, and instead hydrogen ions formed in water are immediately hydrated to form hydronium ions, which are H3O plus. Okay, if you can see the structure. So through proton hopping, the short hops of protons between a series of hydrogen bonded water molecules results in a very rapid net movement of a proton over a long distance, and thus the movement of hydronium and hydroxide ions in an electric field is extremely fast compared to the movement of ions such as sodium and chloride. Okay, if you see over here. So hydronium ion gives a proton, one second, and there is a proton hopping basically. So water accepts proton and becomes a hydronium ion. So the equilibrium constant for the reversible ionization of water is this. Okay. So in pure water at 25 degrees Celsius, that is room temperature, the concentration of H2O is 55.5 molar. On rearranging, the equation becomes this. Okay. So in place of H2O, we have replaced this H2O with 55.5 molar, because we are assuming it's pure water and the temperature is 25 degrees Celsius. So this gives us H plus plus OH minus, which is Kw. So Kw designates the product 55.5 molar into K equilibrium basically, and it's the ion product of water. Now, since the value of K equilibrium determined by electrical conductivity experiments is this thing 1.8 into the power minus 16 molar, the equation becomes this: Kw H plus OH minus equal to 55.5 molar into this, or Kw equal to 1 into the power minus 4, okay, mole square. So the above calculations indicate that the product H plus OH minus in aqueous solution at 25 degrees Celsius always equals this thing. So this is when the concentrations of H plus and OH minus in solution are equal, that is, there is a neutral solution at neutral pH. So if H plus ion is equal to H minus ions, this will be equal. So this will be 1 into the power minus 7 molar. Okay. Yes, somebody. Yes. Okay. I'll share the screen again. Wait 1 second. There must be some issue. Can you see my screen now? Okay. So you're talking about the Henderson-Hasselbalch equation. So the ion product of water Kw is the basis for the pH scale. Okay, and the pH scale is nothing but a very convenient way of designating the concentrations of H plus and OH minus in solution in the range between 1 molar H plus and 1 molar OH minus. So the pH is defined by this equation. Okay, minus log of H plus ion concentration. So you can see this is the pH table basically. So precisely neutral solution. Neutral solution means that the concentration of H plus will be equal to the concentration of OH minus. So when pH is equal to 7, that is when the solution is neutral. Now solutions in which the H plus ion concentration is more than the OH minus ion concentration, that those are acidic, and they'll have pH values less than 7. But solutions in which the H plus concentration is less than OH minus concentration, those solutions are known as basic, and they will have pH values greater than 7. Okay. So if we talk about the pH values of some common lipids, these are the pH values. Okay, if we have kept 1 molar HCl as the standard at this end and 1 molar NaOH as the standard at this end. So you see gastric juice, okay, the gastric juice in your stomach, lemon juice, all of these have a very acidic pH. Cola, vinegar, red wine, beer, black coffee. Neutral, water neutral, human blood, all of these are at neutral pH. Then we have increasingly basic, and we find that household bleach, which is basically sodium hypochlorite, household ammonia, all of these are increasingly basic. Okay, so in these cases, the concentration of hydrogen ions is more than the concentration of the OH minus ions. However, in these cases, hydrogen of OH minus ions is more than the hydrogen of concentration of OH minus ions is more than the concentration of the hydrogen H plus ions basically. And in the neutral cases, both of these are equal. The concentration of OH plus and OH minus ions are equal. So this is basically the pH scale. So these are the pH values of some common aqueous fluids, and we should remember that the pH scale is logarithmic, and the solutions that differ by 1 pH unit actually differ by 10-fold in H plus. So if we say that one solution, the pH of one solution is 5 and the pH of another solution is 4. So even though they differ by 1 pH unit, they actually differ by 10-fold change in H plus ions. Okay. And the H plus ions of a solution of known pH can be calculated by rearranging the pH equation to its exponential form. Okay, so the pH of biological solutions is carefully controlled because pH strongly affects the structure and activity of biological macromolecules. So low pH can, dismantle your, denature your enzymes, proteins, etc. So a proper controlled environment for biological samples is required. Okay. So a little bit about the dissolution constants of weak acids and bases. So unlike strong acids like HCl and H2SO4 and strong bases like sodium hydroxide, potassium hydroxide, there are weak acids and bases such as acetic acid and amino acids, which are not completely ionized when dissolved in water. But instead, depending upon the pH, both the proton donor species, the conjugate acid, and the proton acceptor species, the conjugate base, they occur together in solution. So each weak acid has a characteristic tendency to lose its proton in aqueous solution. The stronger the acid, the greater the tendency to ionize. Okay, so tendency of a conjugate acid to lose a proton and form its conjugate base A minus is defined by the equilibrium constant Keq for the reversible reaction over here. Okay, so equilibrium constants for ionization reactions are usually called acid dissolution constants or Ka. So the higher the value of Ka, the stronger the acid. Biochemists have also commonly converted Ka values to the logarithmic pKa. So the lower the value of pKa, the stronger the acid. Okay, and these are commonly determined by titration of weak acids. Okay, the Ka for weak acid can be calculated from the pKa using the exponential form of the pKa equation. Now, what are the conjugate acid-base pairs of weak acids and bases? So, the dissolution reactions for a number of common mono, di, and triprotic weak acids and bases are shown in this figure, and the pKa values for the dissolution reactions are also shown where they occur along a pH gradient. If you can see over here, this is the pH gradient. We have talked about titration curve in one of the previous classes. This is the titration curve and the pKa basically. Now, titrations are basically experiments that are used to determine the amount of weak acid in solution. And titrations also yield the pKa value for a weak acid. So, in a titration, the conjugate acid form of a weak acid, HA of the weak acid, is stoichiometrically converted to its conjugate base form, A minus, by the addition of a strong base. Okay. So, here the titration curve of acetic acid is shown. So, as shown here, the pKa value of the acid is equivalent to the pH obtained at the midpoint of the titration curve where 50% of the acid has been neutralized. Okay. So these are titration curves of all the weak acids. They are almost have the similar shape. They only differ with respect to where the midpoints of the curves intersect the pH axis of the graphs. And the region in which the buffering capacity is highest for each weak acid corresponds to a pH range where approximately 10% to 90% of the proton donor species has been neutralized. Okay. Now, what are buffer systems basically? Buffers are aqueous solutions that tend to resist changes in pH. So, when small amounts of strong acid or base are added, an optimum buffer consists of equal amounts of the conjugate acid and conjugate base forms of a weak acid in solution. As as shown from the titration curves, pH changes are the smallest at the midpoint of the titration curve, where HA is equal to A minus on addition of an acid or base. So at this point, the pH is equivalent to the pKa of the weak acid. Now, because buffer systems have both HA and A minus species, they are capable of absorbing either strong base or strong acid. Now, whenever acid or base is added to the buffer, there is a small change in the ratio of the conjugate base and the conjugate acid, and there is a small change in the pH. The sum of all the buffer components in solution does not change, only the ratio changes. Now, coming to the equation that we started with. The Henderson-Hasselbalch equation. It's a very popular equation. The shape of the titration curve that we have seen for all weak acids is described by this equation, Henderson-Hasselbalch. This equation simply states the expression for the ionization of a weak acid as shown in the following derivatives over here. Okay. So after solving for H plus, the equation becomes this. Then if you take the negative logarithm of both the sides, and then after substituting the pH and the pK into the equation, it becomes this. So finally, the Henderson-Hasselbalch equation results after mathematical manipulation of the long term on the right, namely this. That is how it happens. Okay. The examination of this equation shows, so when A is the conjugate acid of HA and HA is its conjugate acid. Okay, so Henderson-Hasselbalch equation allows us to calculate pKa given the pH and the molar ratio of the proton acceptor and the donor in solution. Also pH given pKa and the molar ratio of the proton acceptor and donor, and the molar ratio of the proton acceptor and donor given pH and pKa. So we know the equation. So if 3 values are given, we can find out the 4th value basically. Okay, so if 3 values are given, we can find out the 4th value basically. So one unknown we can find out. So this is the Henderson-Hasselbalch equation. Okay. Now, what is the histidine R group of a system? So cells and extracellular fluids contain a very high concentration of proteins. Now, histidine is an amino acid that occurs in most proteins. So the R group or side chain of histidine has an imidazole functional group that undergoes reversible protonation. Now, because the pKa of the imidazole R group is 6, histidine residues in proteins help buffer and pH of the cell cytosol and extracellular fluids around neutrality. And the cell cytosol also is buffered by phosphate, which has a pKa of 6.86. Now, blood is also buffered by the bicarbonate system. Okay, so if we talk about the bicarbonate buffer system, so what happens in that? So in the human stomach and the duodenum, also the bicarbonate buffer system is basically acid-base homeostatic mechanism involving the balance of carbonic acid H2CO3, bicarbonate ion HCO3 minus, and carbon dioxide CO2 in order to maintain pH in the blood and duodenum among other tissues to support a proper metabolic function. So catalyzed by carbonic anhydrase, carbon dioxide reacts with water to form carbonic acid, which in turn rapidly dissociates to form a bicarbonate ion and a hydrogen ion. Okay. So as with any buffer system, the pH is balanced by the presence of both a weak acid, for example H2CO3 carbonic acid, and its conjugate base like HCO3 minus, which is bicarbonate ion, so that any excess acid or base introduced to the system is neutralized. Okay. Now the bicarbonate buffer system is very important in case of cellular, from cellular perspective. It plays a vital role. So in the human stomach and duodenum, this system serves to both neutralize gastric acid and stabilize the intercellular pH of epithelial cells via the secretion of bicarbonate ion into the gastric mucosa. Okay. So in patients with duodenal ulcers, Helicobacter pylori eradication can restore the mucosal bicarbonate secretion and reduce the risk of ulcer occurrence. Okay. So that is the buffer system, bicarbonate buffer system as well. Yeah, so we have the bicarbonate buffer system here also. So it's also an effective physiological buffer near pH 7.4. So this is because the this H2CO3 of blood plasma is in equilibrium with the large reservoir capacity of CO2 in the airspace of the lungs. So the buffer system involves 3 reversible equilibria, in this case between gaseous CO2 in the lungs and bicarbonate ions in the blood plasma. Okay, so this is what makes the bicarbonate buffer system. So when H plus is added to the blood plasma as it passes through the peripheral tissues, this reaction 1 basically shifts towards the production of H2CO3. Okay, and this in turn increases CO2 in the blood. This increases CO2 in the blood as according to reaction 2, and thus increases the partial pressure of CO2 in the airspace of the lungs. Now the extra CO2 is then exhaled. Now when H plus is lost from the blood plasma, the opposite events occur and more H2CO3 dissociates into H2CO3 minus and H plus, and thus more CO2 from the lungs results in the blood. And the rate of inhaling and exhaling, which is controlled by the brain stem in response to pH changes in the blood plasma, can quickly adjust these equilibria to keep the pH of blood nearly constant. The partial pressure of CO2 in the gas phase is denoted by PCO2. So at the pH of the blood plasma 7.4. Yes, yes, say. Okay, okay. Okay. Is anybody okay? I'm sorry, I don't know why is this. Which was the last slide that you could see? Okay. Yeah. If anybody else is trying to screen share, please don't. That is basically, I don't know what is happening. This has not happened the last 2 times. 1 second. Give me a second. I'll just try to see what is wrong. Thank you for letting me know. Just let me check once. Okay. Okay, let me share my screen now. I hope the issue won't happen again. Sorry. Let's just. Yeah. So I hope my screen is visible. Okay. Yeah. So what happens? Yeah. Thank you. So when H plus is added to the blood plasma, as it passes through the peripheral tissues, you can see in this when H plus is added, this reaction shifts this side. So H2CO3 is produced more, and if that happens, then CO2 is also produced more. Okay, so it increases the CO2 level in the blood, and that increases the partial pressure of CO2 in the airspace of the lungs, and the extra CO2 is then exhaled. But when H plus is lost, the exact reverse like opposite events occur. So more H2. So when H plus is lost, more H2CO3 is dissociated into H plus and H2O minus, HCO3 minus, and then more CO2 from the lungs dissolves in the blood. So the rate of inhaling and exhaling, which is controlled by the brainstem in response to the pH changes in the blood plasma, can quickly adjust this equilibrium to keep the pH of blood nearly constant. And it's important that we keep there should be a homeostasis inside our system, right? So it's very important that the pH of the blood remain constant. So the partial pressure of CO2 in the gas phase is denoted as PCO2. Okay, now let's talk about the pH optima of enzymes. So if we know that obviously enzymes are mostly proteins, but RNAs can also be enzymes. But if we talk about enzymes in general, they are biological catalysts. Correct? So they basically accelerate a reaction. So what happens? Every enzyme has a pH optima and has a temperature optima as well. So if we talk about pH, every enzymes they show typically maximal catalytic activity at a characteristic pH, and that we call the pH optimum. So above and below the optimum pH, the activity usually declines. For this reason, it is very important that the pH of the biological fluids such as the cytosol and the blood, they are controlled within a very narrow range. Okay. For example, in untreated diabetes mellitus, high concentrations of ketones or the weak acids accumulate in the blood, reducing its pH. Several ketoacidosis leads to headache, drowsiness, nausea, vomiting, and diarrhea, followed by convulsions, stupor, etc. And these symptoms are caused by the disruption of proper enzyme function at low pH. Okay, so basically what happens when there is a higher level of ketones in blood, which happens in case of diabetes mellitus. Okay, so if a person is suffering from diabetes, if you see their breath smells like acetone. Okay, acetone is also a type of ketone and it's the same fruity smelling substance. Okay, this basically suggests that there are high levels of ketones in the blood. So as the ketones build up, they increase the acidity of the blood. Okay, and that can be really toxic. Okay, so we need to keep the blood pH at a neutral level, otherwise that will turn out to be very problematic for humans. Okay, so that is what happens. There is a high concentration of ketones which accumulate, reducing its pH during diabetes mellitus. Basically it's causing, basically it's making the blood acidic. Okay, and that can lead to a lot of issues. So if you see, this is a graph. This is on the x-axis you have pH, on the y-axis you have percent maximum activity. Okay, so if you see, this is basically at this particular pH conditions, we have wherever you can see the peak, no, at that particular pH condition, yeah, that particular enzyme shows the maximum activity. Okay, so you see trypsin shows maximum activity at around 7 neutral pH, whereas alkaline phosphatase shows maximum activity at around 7 or 8. Okay, trypsin shows around 6.5 to 7. Okay, yeah, alkaline phosphatase shows maximum activity at around 8, 8.5. Pepsin shows maximum activity at 1.5 to 2. Okay, so where do we find pepsin? Can anybody tell me? Yeah. Yes. So a pH, optimum pH of around 2 allows pepsin to operate in its natural acidic environment, while at neutral pH the protein is denatured. So if you want pepsin to act in you know normal pH conditions, like at pH 7 or something, it will get denatured. It will not function. Pepsin is basically a stomach enzyme, correct? That serves to digest the proteins found in the ingested food. So we know that the gastric chief cells in our system secrete pepsin as an inactive zymogen called pepsinogen. And then the parietal cells within the stomach lining secrete hydrochloric acid that lowers the pH of the stomach. And the low pH, like 1.5 to 2, activates pepsin. Correct? So if you want pepsin to function at neutral pH, that is really not possible. And if you want trypsin to act as acidic pH, again that is really not possible. So there are different enzymes which function maximum, show maximum activity at a particular pH, and that is how they should be. Okay, so that was that. We were talking about the Henderson-Hasselbalch equation in the previous question. We have discussed that. So we can move to the next question. So living organisms typically use blank amino acids and dash sugars. What do you think is the correct answer? Okay, anybody else? Yes, so mostly all life forms use L amino acids and D sugars. Okay, so in every life form on earth, sugars are always right-handed and amino acids are always left-handed. Okay, so that, so what does this mean and why does this happen? So this phenomenon involves chiral molecules obviously and is known as homochirality. So we know that chiral molecules are basically molecules of the same formula but with slightly different structures. So your hand is a chiral object because it is not superimposable on its mirror image. So when you place your left hand over your right, your thumbs will point in the different directions. So in organic molecules, when 4 different groups around the central carbon, the molecule will have 2 different chiral forms known as enantiomers, right? So these 2 molecules are mirror images of each other, as the groups around the carbon will be oriented in different directions like left and right hands. Okay, so in chemistry, we can call these R and S enantiomers or isomers, However, when amino acids are sugars or chirals, we basically call them D and L isomers. So D means right-handed, and L means left-handed. So the D and L isomers are normally present in a 50-50 ratio, and when it is present in 50-50 ratio, they are called a racemic mixture. Okay, so naturally one can expect that you'll see them equally present in our bodies, right? But no, in fact, in all life forms, only we use only there are, you can see, L amino acids and D sugars. So the presence of only one as isomer is called homochirality. Okay, so from the smallest organisms to the largest organisms, only L amino acids make up proteins and only D sugars make up polysaccharides. Okay, so in other words, all proteins can be all right-handed and all polysaccharides left-handed. So why is this? So it is quite a mystery, and there are research going. People are doing research on it. Okay, so also it should be noted that some L sugars can also be broken down by metabolic pathways. So sugars can form rings, and the D sugars form a more stable ring than the L sugar. But then L sugars can still exist. The stability of the ring may be the main contributor to the prevalence of the D sugars. Okay, so there are certain theories that people have come up with. Okay, for example, if we talk about enzymes and evolution, so the enzymes in cells can only metabolize one isomer but not the other. Okay, enzymes catalyze reactions to break down and build molecules by binding to them, right, in very specific ways. So an enzyme that breaks down D-glucose will probably not be able to break down L-glucose. So if you wanted to use and build both isomers, we would have to have two sets of enzymes, correct? So because one set of enzyme will not be able to break two types of glucose, D-glucose and L-glucose. So if you wanted to use and have both isomers, we would need two sets of enzymes. So this would require much more DNA, more food, and a lot more energy. So naturally, cells want to be as efficient as possible. So having an extra set of enzymes can be, taxing. Okay. But if we had those extra enzymes, couldn't we also get twice as much energy from the environment or our surroundings? So we could use the other 50% of isomers if we had the enzymes to break them down instead of wasting them. Okay, so people have come up with various theories. So apparently, when early life was evolving, an organism would have enzymes that selectively react with one isomer. Okay, so the organisms that ate this first life form would have to have the same selective enzymes in order to digest them. So if all your food only contains D sugars, so will you. So if the bottom of the food chain only uses L amino acids, homochirality will spread to every organism in the food chain, right? And since life had to develop one set of enzymes first, I think we wound up being stuck with that selectivity. So selective pressure from evolution forced each new organism to use the same amino acid and sugar types as the ones before it. So once one isomer was picked, every form of life will use it. But then why L amino acids instead of D amino acids, and why D sugars instead of L sugars? So that is the real mystery. Why something at the beginning of life had to select one over the other. So it is possible that in the early stages of life, some organisms developed one set of enzymes while different organisms had the other, and it could have been a very random event that wiped out one line of enzymes. But scientists aren't really, not everyone is satisfied with this answer. So there has to be some proper reason why L amino acids and D sugars are better for survival than their counterparts. So obviously there will be other explanations as well with respect to physics, chemistry. So we discussed the evolutionary part of it, which is obviously biological explanation, but there should be other explanations based on polarized light and based on physics basically. And if we talk about from the chemistry perspective, there might be other explanations based on racemic mixture and all of that. So we'll not go into too much detail for now, but for now, okay, we'll say that there were L amino acids and D sugars. But however, in recent research papers, it has been seen that D amino acids were actually recently found in some various living higher organisms as well, in the form of free amino acids and peptides and proteins. Okay, and free D-aspartate and D-serine are apparently present and may have important physiological functions in mammals. Okay, so that was seen in one of the papers in the last 1 or 2 decades. So maybe that can lead to new studies in the future. Okay, so for now we'll just remember that living organisms typically, so the typically word is very important, typically use L amino acids and D sugars. Okay, so we'll talk about the D and L notations briefly. So they are used to describe the configurations of the carbohydrates and amino acids. So glyceraldehyde has been chosen as the arbitrary standard for the T and L notation in sugar chemistry because this is an asymmetric carbon and can exist as a pair of enantiomers, if you see over here. So in Fischer projection, the carbonyl group is always placed on the top position for monosaccharide, and from its structure, if the OH group is attached to the bottommost uh, asymmetric carbon, that is the carbon that is second from the bottom, it is on the right, then the compound is a D sugar. Okay, and if the OH group is on the left, then the compound is an L sugar. Okay, and almost all sugars found in nature are D sugars. Okay. So like R and S, D and L indicate the configuration of an asymmetric carbon, but they do not indicate whether the compound rotates polarized light to the right or to the left. For example, D-glyceraldehyde is desterotatory, whereas D-lactic acid is levorotatory. So do not confuse D-glyceraldehyde to be going with this D thing. Okay, that is not this D and L configure. This D and L notation, they do not indicate whether the compound rotates polarized light to the right or to the left. Okay, we have an example like D-glyceraldehyde is desterotatory, whereas D-lactic acid is levorotatory. So in other words, optical rotation like melting point or boiling point is a physical property of a compound. Okay, but R, S, D, L are conventions that scientists have started to indicate the configuration of a molecule. Okay, so if we talk about the ketose and aldose D and L configuration, this is L glucose, this is D glucose. Okay, you can see that the only difference between these two is how the OH and the H are arranged. If you can see, it's exactly opposite in case of D and L glucose. Similarly, for D and L fructose is basically we have a ketone group over here, we have an aldehyde group over here, right? So you can see how the ketone, fructose and glucose have the D and L notations. Now, what are epimers? Epimers are basically stereoisomers that differ in configuration of only one asymmetric carbon of enantiomers or diastereomers. For example, D-glucose and D-mannose are C2 epimers. You see, everything else is same. Only the directions of the H and OH attached to the C2 carbon is different. So those are epimers. Okay, whereas D-glucose and D-galactose are C4 epimers because in this case the components attached to the C4 carbon differ in the directions. Okay, whereas D-fructose and D-tagatose are C4 epimers of ketohexose. Okay, so what is the mechanism of epimerization? So in the presence of a base, D-glucose may be converted to D-mannose. Okay, via the removal of hydrogen at C2 carbon followed by the protonation of the enolate. So if you see this OH will go and attack over here, and this is a double bond forms over here, then there will be ultimately a release of there will be a removal of hydrogen, and ultimately D-glucose can be converted to D-mannose. So D-glucose and D-mannose, that they are C2 epimers. So this is how the epimerization reaction can happen. Okay. Now, what are anomers? Anomers is basically a stereoisomer having different configuration at the anomeric carbon. So it is the type of geometric isomer found at certain atoms in carbohydrate molecules. So you see, the anomeric carbon is basically the carbon derived from the carbonyl carbon, the ketone or the aldehyde functional group. So whatever the functional group is, be it ketone or aldehyde, so that is our carbonyl carbon, right? So the anomeric carbon is that carbon that is derived from the, so in here this is the anomeric carbon, right? So the anomeric carbon is the carbon derived from the carbonyl carbon from the functional group of the open chain of the carbohydrate molecule. And the anomerization is basically a process of conversion of one anomer to another. Okay, so in this case, which one do you think is the anomeric carbon? C1. Okay, C1 means the one label that we have seen over here. So in here you see, in case of alpha D-glucopyranose and beta D-glucopyranose, the only difference is that in this case H is here, OH is here, as in this case in the beta case OH is above and H is below. So these are different nomenclatures and different notations, and we need to know what each of these mean. So this is the mechanism of anomerism. Okay, so the cyclic structure of sugars are usually favored. Hemiacetals in acquisition and equilibrium with their open chain forms. So this is the entire mechanism of anomerization. We will not go into too much detail. However, anomerization basically occurs to the protonation of the cyclic acetyl oxygen, ionization to form an oxocarbenium, okay, with release of an alcohol, and nucleophilic attack by an alcohol on the reverse face of the oxocarbenium ion, followed by deprotonation. This is the entire structure of mechanism of anomerization. Okay, if we move to amino acid, that means protein. Basically, proteins are obviously more than energy source. We have proteins, we use proteins as receptors, even hormones, even cytokines, then signaling molecules, enzymes, etc. Okay, so polypeptides are basically shorter, like less number of amino acids forming less number of peptide bonds. For proteins, they are obviously much huge structures. So what are the building blocks of protein? Those are the amino acids. Amino acids, they have a carboxyl group at one end and an amino group at another end. In case of proteins, with amino acids, we have these four components. So they have a carboxyl group and an amino group, and all the amino acids from protein have the NH2 attached to the C alpha to the COH. Okay. Now all naturally occurring alpha amino acids except glycine are chiral and the L stereoisomer. Can someone tell me why glycine is not chiral? Anyone? Correct. So the carbon is basically not asymmetric. We need four different components to be attached to the C alpha carbon. But in case of glycine, the R group is H only. It's the simplest amino acid. The R group is also H. So there are two H groups in glycine. So that is why glycine is not chiral. Apart from glycine, every other amino acid, they have a different R group. So every other amino acid, they have a different R group that makes all the four components very different to each other. Okay, so that's why all naturally occurring alpha amino acids are chiral and the L stereoisomer. Okay, so there are 20 standard alpha amino acids in naturally occurring protein, and by convention the NH2 is placed to the left. Okay, and if you see, so we have talked about amino acids and how they can be categorized before. So if you have non-polar R groups which are hydrophobic, these are those. If we have the polar R groups which are hydrophilic, water loving, then we have these structures. And then we have, so we have already talked about all these. Okay, so among the aromatic amino acids, tryptophan and phenylalanine belong to this category, whereas tyrosine, because of this OH, it can belong to this polar R group hydrophilic category. Now we know that amino acids contain an acidic functional group COH and a basic one. Okay, so thus reactions are highly pH dependent. Okay. We have talked about zwitterionic properties. Zwitterion ions, basically when there are equal amounts of positive and negative charges. Okay. So this for different enzymes, for different amino acids, this will be different. So this is a leucine zwitterion, because both of them are in ionized form, COO minus and NH3 plus. So we have talked about the basic definition of zwitterions. Zwitterions are basically it's basically a molecule that contains an equal number of positively and negatively charged functional groups. Okay, so that is what this zwitterion is. So how are peptides linked to each other, and how is the peptide? Sorry, how are amino acids linked to each other, and how is the peptide bond formed? So think about these two amino acids, alanine and glycine. Okay, so in case of alanine and glycine, they'll form a peptide bond between themselves with the release of H2O, dehydration hydrolysis, and see, so this is the peptide amide linkage. This will be the N terminus, and this will be the carboxylic end or C terminus. This is a dipeptide because there are two amino acids forming one peptide bond, so dipeptide. Okay, so this is what mean two amino acids, one peptide bond. tripeptide will be having 3 amino acids. Okay, so if we take glycine, alanine, and phenylalanine and want to join them, link them, we can form tripeptides with 2 peptide bonds and 3 amino acids. Okay, so a little bit about the levels of protein structure. So protein will have a primary structure, obviously, which is basically the sequence of amino acids in the peptide chain and the location of the disulfide bridges. So basically, mainly the sequence of amino acids, that's a primary structure. So that is derived from the from DNA basically. Whatever information was stored in the DNA was passed on to the mRNA via transcription, and then from mRNA to protein via translation. So the primary structure of protein will be the sequence of amino acids that was read from the DNA, right? And then we have the secondary structure, which is basically a description of the conformation or shape of the backbone of the protein. They can take alpha helix, beta shape, all these secondary structures, turns, loops. The alpha helix and beta sheets are the most popular and frequent. Then we have the tertiary structure, which is basically a description of the 3D structure of the entire polypeptide. How the proteins exist in 3D level. And if the protein has more than one chain, that is, we know that there are some proteins which have more than one subunits, right? So subunits need to come together, fold with each other and upon each other. So if a protein has more than one subunit chain, it can have a quaternary structure as well. Okay. So there are some protein sequences. Basically, these are hormones. Hormones made up of amino acids, as you can see from here. Insulin. So secondary structure is basically it's a fixed arrangement of amino acids resulting from interactions between amide linkages that are close to each other in the protein chain. Interactions can be hydrogen bonds, okay, and many H bonds are sufficient to define that shape. So what can be the ionic interactions in proteins? There can be salt bridges that will involve CO minus and other NH3 plus groups. Along with H bonding and dispersion forces, these are responsible for the overall shape or conformation of the protein. Okay, this can be the secondary structure. So you can see there will be hydrogen bonds sheets. So they can be alpha helix. You can see their H bonding is intramolecular. I think I and I plus 4, one amino acid, and the fourth alternative amino acid can have an H bond in case of alpha helix. Now, what is the tertiary structure of proteins? Basically, they arise from the weaker attractive forces, which are non-polar dispersion forces between the hydrophobic parts of the same chain that are widely separated in the primary structure, but they are close in space. Okay, so these result in chain twisting and folding. So, the primary sequence of protein is very important, obviously, because they are being dictated by the DNA. Okay, so if there is any kind of mutation in the DNA, if there is a deletion, insertion, or there is a change in the frame of reference, any kind of mutation in the DNA, that will basically change the amino acid sequence, correct? Because we take every three nucleotides basically to form one codon, and that will give rise to one amino acid. So if even one is deleted or inserted, that will change our frame of reference. Okay, now the primary sequence of proteins is important, but the 3D structure is equally important, because without proper folding, without proper structural rearrangement, none of the proteins will be functional. Okay, for example, we have a protein, but its structure, we have the exact polypeptide sequence, okay, but the structure has not folded into its native conformation, so it will be non-functional. It will never do its job. It has denatured basically. So will it do its job? No, it will not be functional in biological context. That is why the 3D structure of the proteins is important. Okay, so there can be dispersion forces where attractive and nuclei are separated by the sum of their van der Waals radii. Okay. So tertiary structure of protein, it can be collagen, a fibrous protein that has a triple helix structure. We can have hemoglobin, which is a globular protein basically. So you see, this is the collagen, which is a triple helix structure. Hemoglobin, which is basically will form a globular structure. Okay, 1 second. Moving on, we know about active site of enzymes, right? So active site are also made from proper folding of the proteins only. So if my protein is not folding properly, I will not find the native active site which is required for the protein to carry out its enzymatic functions, right? So the active site of enzyme is also very important. Now denaturation. So any physical or chemical process that changes the protein structure and makes it incapable of performing its own normal function is called denaturation. So whether denaturation is reversible, it depends on the protein and the extent of denaturation. So for example, we have if we heat egg whites, that will be irreversible denaturation. If we are waving of hair, that can be permanent denaturation. So proteins by structure, if we take, we can have simple or conjugated structure. Okay, lipoprotein, glycoprotein, hemoprotein. And if we are only talking about the structural components, only without any kind of conjugation with anything else, then we can have fibrous or globular structures. Okay, so fibrous structure can be collagens, elastins, keratins, myosins, all of these. Globular structure can be these. Okay, proteins by function, obviously enzymes will be acting as biological catalysts. They can be contractile, muscles, hormones, neurotransmitters. We know proteins have a lot of functions. They're very diverse in their function. So if you talk about the non-essential amino acids, basically why do we call them non-essential and why do we call them essential? So the non-essential amino acids can be synthesized in our body. Okay, so why is it non-essential? That means because these are non-essential in our diets. It's not required in our diets because our body can synthesize those on its own. But there are essential amino acids that our bodies cannot synthesize on its own. So they are essential in our diets. So they are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, and even arginine in infants. Okay. So proteins are well defined. They can be coded precisely by genes, stranded peptide link. Okay, whereas polysaccharides, they are synthesized by enzymes without template. There are many homopolymers. They have various links. It's not for proteins. There are only standard peptide link. But in case of polysaccharides, they can be alpha 1,1, alpha 1,2, beta 1,4. There are different types of polysaccharide link. Okay, so the glycosidic bond that will form between the two monosaccharides. So the monosaccharides can contain between 3 and 7 C atoms. Okay, they can be glyceraldehyde, which is a triose. Why triose? Because C carbon. Okay, they can be erythrose, 4 carbon, tetrose, ribose, 5 carbon, pentose, glucose, 6 carbon, hexose, fructose, also an hexose. But this is an aldohexose, this is a ketohexose because glucose has an aldehyde group, whereas fructose will have a ketone group. Okay, so these are monosaccharides with various numbers of carbon atoms. We have talked about asymmetric chiral carbon that has a covalent bonds to 4 completely different groups. So that is why glycine was not chiral, because glycine had 2 similar groups, 2 hydrogen attached to the C alpha group. So that made it the simplest amino acid, and so apart from glycine, everything else was chiral. Okay, we have talked about all of these different types of forms. 3 examples of chiral carbon atoms. We have talked about this. Ring formation. This is alpha D glucose, aldose. This is how ring formation can happen. Okay, ketose also similar. So the ring structure, linear form can be shown as something known as Fischer structure, and the ring form can be shown as something called the Haworth's projection. We'll talk about these also in one of the further questions. Now cyclization can happen via intramolecular hemiacetal or hemiketal formation. Okay, and the C1 becomes chiral upon cyclization. C1 is the anomeric carbon, that is the carbon in the functional group, the aldehyde of the keto functional group. Now anomeric C contains OH group, which may be alpha or beta. Okay, so these are 2 different forms of beta D glucose. Okay, so they can be an equatorial substituent or an axial substituent. Okay, So how the formation of di and polysaccharide bonds. So dehydration synthesis of a sucrose molecule formed from condensation of a glucose with a fructose. Okay. So in this case there is a 1-2 linkage. Okay, 1 carbon 2 carbon 1-2 linkage is happening. For lactose there is a beta 1,4 glucose linkage. You see how there is one galactose unit, one glucose unit that will form lactose. For maltose there is alpha 1,4 linkage happening, one glucose unit, one glucose unit, two of them coming together forming maltose. Disaccharides are basically composed of monosaccharides units by glycosidic link from C1 of one unit to the OH of the second unit. So these can be the most common units. Links can be alpha or beta, and the link around glycosidic bond is fixed, but anomeric forms on the other CR1 are still in equilibrium. Okay, so polysaccharides. So these can be linear polysaccharides like amylose, which I told you already, which we have discussed in one of the previous sessions. It is one form of starch. Amylose, cellulose, then ululan. Alternating repeat can be agarose. Interrupted repeat can be pectin, gitosan. Block copolymer can be alginate. Branched will be amylopectin, dextran, lignin. And complex repeat can be xanthan. Okay. Wait 1 second, this is a network issue. Okay, so you have already talked about the different conformations. Okay, don't need much detail. So yeah, so the previous question was amino acids and glucose, sugars. So in general, typically in nature we see L amino acids and T sugars. Okay, that's what we talked about. Now, next question we are coming to. This is the next question for today. Which of the following statements is false with respect to illustrations of sugars? Fisher projections are typically used for open chain forms. Howard projections are typically used for cyclic forms. Fisher projections are typically used for cyclic forms, and Howard projections are typically used for open chain forms. Anybody? We discussed it just. Okay, it's a wrong statement. Cyclic forms. Yeah. So, so if we discuss more about the Fisher and the Howard projections, so this is Fisher projection basically shows the chirality of the organic molecules in the open chain form and doesn't use dashes and wedges. So horizontal axis points out of the plane towards the viewer, whereas the vertical axis, if you are looking from this point, vertical axis points back into the plane away from the viewer. Okay. So to determine whether each carbon of a Fisher projection is S, R and S, we basically assign a priority to the 4 substituents, right? So the first will assign a priority, and then we'll face the hydrogen back away from the viewer, and then we'll determine whether it is clockwise or anti-clockwise, counterclockwise. So if it's counterclockwise, it will be R, if you can see over here. Okay, so when the hydrogen atom or whatever group is lowest priority is on the horizontal axis, the molecule must be rotated on its vertical axis to swing the hydrogen to face away from the viewer. Okay, so it's wise to determine whether each chiral carbon is R or S to be sure that the two types of diagrams are basically represent the same molecule. So this is a Fisher projection. You can also write in this form, or in this form, line Fisher, and in a zigzag form as well. Now the Howard diagram shows stereochemistry like a Fisher diagram. Okay, so this is a light structure. This is the Howard projection. This is a chain conformation. Okay, so both chair and board conformations, they show conformation and stereochemistry. Okay, so if we talk from sugar like carbohydrate perspective, aldehyde or ketone groups with multiple hydroxyl groups are carbohydrates. Ketones, both groups attached to the carbonyl group are carbon. Aldehydes, one carbon and one hydrogen are attached to the carbonyl group. So this is an aldehyde group and this is a ketone group. So this is acetaldehyde, this is acetone. See, I've talked about monosaccharides, which are basically polyhydroxyaldehydes or polyhydroxy ketones. Okay, and these are stereoisomers with identical functional groups linked in the same order but with different spatial orientation. And enantiomers are nothing but having one chiral carbon, so they are non-superimposable mirror images. So this is a line structure of D-glucose. So in Fischer projection, there are straight chain compound bonds with the vertical bonds representing bonds pointing back and horizontal bonds pointing forward. Okay, so this is how D-glyceraldehyde basically looks. So what are Fischer projections? Using these rules, the distinguishing features and 3D structure of stereoisomers can easily be represented with 2D drawings. So the Fischer projection, named after the scientist Emil Fischer, who established the molecular structures of many sugars. Okay, so this is the Fischer projection. If you see in linear form, this is alpha D glucose and alpha L glucose. Okay, in the linear formats. So this is Fischer projections of all the monosaccharides that we talked about. So these are all the monosaccharides. Glucose is an aldehyde, fructose is a ketone, galactose is an aldehyde, mannose also an aldehyde, and glucose is also an aldehyde. Only fructose here is a ketone structure. So these are the Fischer projections. Okay, aldopentose. This is an aldopentose. Obviously there are 1, 2, 3, 4, 5 carbons. So it's a pentose, and obviously it's a aldehyde group. So aldopentose, D-ribose. Okay, this will be 1, 2, 3, 1, 2, 3. So this will be a triose because there are 3 carbon and obviously an aldehyde group. So aldotriose. Okay, now many monosaccharides, most commonly pentoses and hexoses, they have cyclic structures in aqueous solution in which the carbonyl group has formed a covalent bond with the oxygen of a hydroxyl group around the chain. Okay, so if you see over here, so D-glucose can form a D-glucopyranose. Okay, so those were the Howard projections. So cyclic structures depicted by 3D drawing involve a set of rules. So carbon atoms are not explicitly drawn. The ring perpendicular to the plane of the paper and heavy line projects towards the reader. So Howard projection was named after Walter Howard, who confirmed the structures of many complex sugars. Okay, so these are the carbohydrate ring structures. So most common 5-membered ring is furanose. Furan is derived from the furan ring. Furan 4-carbon ring with 1 oxygen. So the OSE ending indicates multiple hydroxyls. And most common 6-membered ring is the pyranose, which is the pyranose derived from the pyran ring. It is a 5-carbon ring with 1 oxygen. So this is a pyranose ring structure. If you see, this is the aldehyde alcohol hemiacetal. So an aldehyde can react with alcohol to form a hemiacetal, and a similar reaction can occur intramolecularly in monosaccharides where the aldehyde or the ketone groups react with one of the hydroxyl groups in the same molecule. So then if you talk about the cyclic Howard structures, there are stable cyclic hemiacetals formed. So when the CO group and the OH group, OH are part of the same molecule. So for hexoses, the hydroxyl group on C5 reacts with the aldehyde group or ketone group, and the cyclic structure of a D-isomer has the last CH2OH located above the ring. Okay, so how do we draw the cyclic structure for all these monosaccharides? So you have to number the carbon chain and turn clockwise to form a linear open chain. Okay, we just label the carbons and then we turn it clockwise. Then the chain was bent. Okay, then the chain was bent to make a hexagon. So the bond C5, okay, C5O to C1. So place a C6 group above the ring. So write the OH groups on the C2 and C4 below the ring, and write the OH group on the C3 above the ring. Okay, and write a new OH on C1. So the new OH on C1 is drawn. So down for the alpha anomer and up for the beta anomer. So that is alpha D glucose and beta D glucose. Okay, so bonds pointing right in the Fischer projection are equal to the bonds pointing down. So if you correlate these two projections, if you see that the bonds that were pointing right, okay, in the Fischer projection are all the points that are pointing down. Okay, that is how you can correlate the Fisher and the Howard projections. Okay, so in case of pyranose ring summary, this is a D-glucose open chain form. This is alpha D-glucopyranose, beta D-glucopyranose. Okay, so what happens is the C1 aldehyde reacts with the C5 hydroxyl to form the intramolecular hemiacetal, if you can see over here. So monosaccharides and disaccharides we talked about. Carbohydrates are the most abundant biomolecules on Earth, and each year photosynthesis converts more than 100 billion metric tons of CO2 and H2 into cellulose and other plant products. And carbohydrates are polyhydroxy aldehydes and ketones, or substances that yield such compounds on hydrolysis. So many but not all of them have the empirical formula CH2ON, but some also contain nitrogen, phosphorus or sulfur. So carbohydrates occur in 4 main size classes: monosaccharides, disaccharides, oligosaccharides and polysaccharides. And the most abundant monosaccharide in nature is D-glucose, which is also known as dextrose. So common disaccharide sucrose consists of the 6 carbon sugars D-glucose and D-fructose, and common polysaccharides include cellulose and starches. So both of these are homopolymers of D-glucose units, but with different linkages between the residues. So more complex carbohydrate polymers attached to a protein or lipid moiety are also prevalent in nature. Okay, monosaccharides are colorless crystalline solids that are very soluble in water but insoluble in nonpolar solvents, and most have a sweet taste. So the backbones of the common monosaccharides are unbranched carbon chains in which all the carbons are linked by single bonds. So in this open chain form, one of the carbon atoms is double bonded to an oxygen atom to form a carbonyl group, and each of the other carbons that has a hydroxyl group. So if the carbonyl group is at an end of the carbon chain, that is in an aldehyde group, the monosaccharide is called an aldose, and the carbonyl group is at the other position. In case of a ketone, the monosaccharide is a ketose. So monosaccharides with 3, 4, 5, 6 and 7 carbons in their backbones are called trioses, tetroses, pentoses, hexoses and heptoses. We have discussed already. And many of the carbon atoms to which the hydroxyl groups are attached are chiral centers. So this gives rise to the many stereoisomers found in monosaccharides. So these are obviously the common monosaccharides. The simplest monosaccharides are the 3 carbon trioses. This is a aldehyde, this is a ketone, diglyceraldehyde and aldotriose, and dihydroxyacetone. It is a ketotriose, and the most common monosaccharides in nature are the aldohexose D-glucose and the ketohexose D-fructose. So the aldopentoses D-ribose and 2-deoxy D-ribose are the components of nucleotides and nucleic acids. Okay. I have talked about the D and L stereoisomers. Okay, so all the monosaccharides except dihydroxyacetone contain one or more asymmetric carbon atoms and thus occur in optically active isomeric forms. And the simplest aldose glyceraldehyde contains one chiral center, which is the middle carbon atom, and therefore has 2 different optical isomers or enantiomers. And one of the 2 enantiomers of glyceraldehyde is by convention designated the D isomer and the other as the L isomer. Okay. So in general, a molecule with n chiral centers can have 2 to the power n stereoisomers. Okay, so glyceraldehydes are 2 to the power 1 chiral center, so n equal to 1, so 2. Aldohexoses with 4 chiral centers have 2 to the power 4, n equal to 4, so 16. So the stereoisomers of monosaccharides of each carbon chain length are divided into 2 groups that differ in the configuration about the chiral carbon that is the most distant from the carbonyl carbon. So those in which the configuration of this reference carbon is the same as that of the diglyceraldehyde so diglyceraldehyde is taken as an arbitrary standard, as we talked about before. So in which the configuration of the reference carbon is the same as that of the diglyceraldehyde, they are known as D isomers, and those with the same configuration as L-glyceraldehyde are known as the L isomers. Okay. So the structures of D monosaccharides, we have already talked about all the structures of D monosaccharides. So these are the structures of D aldoses. Okay, 6 carbons, the 3, 4, 5 and 6 carbons. All the aldoses, these are the ketoses, 3, 4, 5 and 6 carbons. Okay. So what are epimers of D aldohexoses? The 2 monosaccharides that differ only in the configuration around 1 chiral carbon atom are called epimers. So D glucose and D mannose are epimers which differ in the configuration at C2. Okay, we have already talked about this. and D-glucose and T-galactose are C4 epimers because they differ in the C4 region. So some sugars are also known to occur naturally in their L form. Okay, so some example can be L-arabinose. Okay, and the L isomers of some sugar derivatives that are common components of glycoconjugates. We have talked about the formation of different hemiacetals and hemiketals. So aldotetrosis and all monosaccharides with 5 or more carbon atoms can occur predominantly as cyclic ring structures, in which the carbonyl group can has formed a covalent bond with the oxygen of a hydroxyl group along the chain, and the formation of these ring structures is a result of a general reaction between alcohols and aldehydes or ketones to form derivatives called hemiacetals or hemiketals. So actually what happens to molecules of an alcohol can add to a carbonyl carbon. So the product of the first reaction for an aldose is a hemiacetal, and the product of the first reaction for a ketose is a hemiketal. So if the OH and the carbonyl groups are from the same molecule, the 5 or 6 membered ring results. The addition of a second alcohol molecule produces the full acetal or ketal, and the bond formed is a glycosidic linkage. So you see we have talked about the cyclization of D-glucose. So the reaction of the first alcohol with an aldose or ketose, they can basically create an additional chiral center at what was the carbonyl carbon. So because the alcohol can add to the carbonyl carbon by attacking either from the front or the back, the reactions can produce either of the two stereoisomeric configurations denoted alpha and beta. For example, D-glucose exists in solution as an intramolecular hemiacetal, in which the free hydroxyl group at the C5 has reacted with the aldehyde C1, rendering the latter carbon asymmetric and producing the two possible stereoisomers alpha and beta. And these two isomeric forms, which differ only in the configuration about the hemiacetal carbon atom, are called the anomers, and the carbonyl carbon is called the anomeric carbon. Okay, and the same type of nomenclature or terminology basically is used to describe the anomeric forms of hemiketals, such as formed by the fructose. So the alpha and beta anomers of D-glucose interconvert via the linear form in aqueous solution by a process called mutarotation, and in solution and equilibrium mixture forms, which consists of about 1/3 alpha D-glucopyranose, 2/3 beta D-glucopyranose, and the trace amounts of the linear and the 5-membered glucofuranose ring forms. We have talked about pyranoses and furanosis. So 6-membered monosaccharide ring compounds are called pyranoses because they resemble pyran, and 5-membered monosaccharide ring compounds are called furanoses because they resemble furan. The systemic names of the two ring forms of D-glucose are therefore alpha D-glucopyranose and beta D-glucopyranose. So the ketohexoses such as fructose also occur as cyclic compounds with alpha and beta anomeric forms. So in these compounds, the hydroxyl groups reacts with the keto group at C2, forming a furanose ring containing a hemiketal linkage. So D-fructose apparently forms a furanose ring very easily, and the more common anomer of this sugar in combined forms or in derivatives is beta D-fructofuranose. We have talked about Fischer projection and Howard perspective formulas. Okay, this is a Fischer projection. This is a Howard perspective formula, which was basically what the question is all about. So cyclic sugars are more accurately represented in Howard perspective formulas than in Fischer projections that are used for linear sugar structures. So in Howard formulas, the 6-membered ring is tilted to make its plane almost perpendicular to that of the paper, and the bonds closest to the reader are drawn thicker than those farther away. And to convert the Fischer projection formula of any linear T-hexose to a Howard perspective formula, the 6-membered ring is drawn, and then carbons are numbered in a clockwise direction, beginning with the anomeric carbon. Then the hydroxyl groups are added. So if a hydroxyl group is on the right, as I said, right in the Fischer projection, it is placed down in the Howard formula. Okay. So similarly, if a hydroxyl group is placed left in the Fischer formula, they are placed up in the Howard projection. Okay, and the terminal CH2OH group projects upward from the D enantiomer and downward from the L enantiomer. And when the hydroxyl group on the anomeric carbon of a D-hexose is on the same side of the ring as C6, the structure is by definition beta. And when it is on the opposite side of C6, the structure is alpha. Okay, so these are some examples. Conformational, so these are the two possible chain forms of beta D-glucopyranose. We will not go into very detail. These are the important hexose derivatives. Okay, so there are many sugar derivatives in which hydroxyl group in the parent compound is replaced with another substituent or a carbon atom is oxidized to a carbonyl atom, carboxyl group. So in addition, these are some of the, we can see, beta D-glucosamine, then glutamic acid, okay, and acetyl beta D-glucosamine, and acetyl glutamic acid. So you can try and check how the structures differ from each other. For amino acids, you have beta D galactosamine, beta D amino sugars, sorry, beta D manosamide. For deoxy sugars, we have base rhamnose, alpha L rhamnose. You have beta D glucuronate, D-glucono, delta lactone, and acetyl glutamic acid, sialic acid, basically an acidic sugar. So okay, so a little bit about the measurement of blood glucose level. So monosaccharides can be oxidized by relatively mild oxidizing agents such as cupric iron, which oxidizes the carbonyl carbon to a carboxyl group. Glucose and other sugars capable of this reaction therefore are called reducing sugars. So this reaction we have talked about Felling's reaction in the previous session, if you remember. This reaction, Felling's reaction, was used for many years to detect and monitor glucose levels in people with diabetes mellitus. However, today there is an immobilized enzyme called glucose oxidase on a test strip, which is much more efficient and is used to catalyze the oxidation of free glucose to D-glucono delta lactone, as you can see from here, and the hydrogen peroxide produced is as the second product of this, is a byproduct of this reaction. Subsequently reacts with the colorless compound in the strip via the enzyme peroxidase to form a color product which can be quantified using a simple photometer. Okay, will not go into too much detail. Intro to polysaccharides. Obviously, so most carbohydrates found in nature can occur as polysaccharides. Okay, that's polymers of medium to high molecular weight. Polysaccharides, also known as glycans, differ from each other in the identity of their recurring monosaccharide units, in linking the monosaccharide units, and in the degree of branching. So like we know, amylose is a linear form of starch, but amylopectin is a branch form. So homopolysaccharides contain only a single monomeric species, whereas heteropolysaccharides contain two or more different kinds. So unlike proteins, polysaccharides generally do not have defined molecular weights. This is because polysaccharides are now synthesized from a template. So we know that proteins are synthesized from a template, right? But polysaccharides are not really synthesized from a template. So they do not have a defined molecular weight. Instead, there is no specific stopping point for the enzymes involved in their biosynthesis. Okay. We have talked about disaccharides and how disaccharides are named. The linkages between two different disaccharides, like for lactose, it's beta D galactopyranose and beta D glucopyranose with the beta 1,4 linkage. For sucrose, it's a beta D fructofuranose with the alpha D glucopyranose with alpha 1,2 linkage. For trehalose, it's a alpha D glucopyranose with another alpha D glucopyranose with alpha 1,1 linkage. That is trehalose. Okay. So you have talked about starch in the first session. We talked about amylose, which is a linear polymer of D-glucose residues that are carried by alpha 1,4 linkages. For amylopectin, it's a branched polymer of D-glucose residues that can weigh up to even 200 million Dalton. Okay. This is how the starch, different categorization of starch can be done. Glycogen also we have talked about. They are very their main linkage is formed between D-glucose units alpha 1,4. For the linear ones, and for the chain ones, they can be at the alpha 1,6. So both linear and chain. Okay. And glycogen is especially abundant in the hepatocytes of liver. Okay, and they can be observed in cytoplasm of cells also by electron microscopy. Then we have cellulose, which is a linear homopolysaccharide composed exclusively of D-glucose units held together by beta 1,4 linkages. Okay, it's found in the cell walls of plants and everything. Vertebrate animals like us, we lack the hydrolytic enzymes cellulase that can cleave the beta 1,4 linkages between glucose units in cellulose. Okay, so these enzymes are produced by many cellulitic microorganisms, but we are not able to produce this. So similarly, cellulases produced by microorganisms living in the humans of cattle, sheep, and goats allow these animals to obtain energy from cellulose present in the soft grasses in their diet. Okay, but I don't think we'll be able to digest cellulose. Okay. Also, we have chitin, I guess. I'm not really sure about how it's pronounced. Chitin only, I guess. Chitin is a Horelian homopolysaccharide. It's composed of an acetyl glucosamine residues, obviously a derivative in beta 1,4 linkage. Okay, it forms the hard exoskeleton of these arthropods. Okay, so the main difference between chitin and cellulose is the replacement of the hydroxyl group at C2 with an acetylated amino group. So chitin also forms extended fibers similar to those of cellulose. So the main purpose of chitin is it's found in the hard exoskeletons of these arthropods. Okay. We have talked about all these things. So this was the last question for today. We have talked about all these structures and the roles of all these polysaccharides, amylose, amylopectin, glycogen, cellulose, chitin, and all of these. The repeating unit, they are linked by alpha 1,4 or beta 1,4 linkages, and what are the two monosaccharides that are linked together. That is very important for all these polysaccharides, and obviously their roles and significance. What are the applications of these polysaccharides? Where are they seen? What are their purposes, and why not this and why cellulose? So cellulose is not that important for us because we are not able to digest cellulose because we don't have the enzymes required for it, but they are the structural components in so many plants that gives rigidity to the strength of the cell walls. So these are the different types of polysaccharides that can be seen in nature. Okay, so today we'll stop here. If you have any doubt, you can ask me, or we'll stop the session over here. And anyone? Yes. Yeah. So the lecture. Lecture slides. You mean of the course instructor, or the lecture slides that we discuss in the live sessions? Those will be uploaded. So the videos will be uploaded on YouTube, and the lecture slides link will be shared by the NPTEL staff. So you don't have to worry about that. Are not shared. Okay, so you check again. I think that has been shared now. You check again and let me know if it's not shared by the next class. Then I'll look into it because we have shared, and the videos have also been uploaded on YouTube. So you check again. If it's not there, then we'll check again. Okay. Thank you. Anybody else? If you don't have any doubt, we can. I'll stop screen sharing for today. So if nobody has a doubt, we can stop the session, conclude the session for today, and maybe meet next Monday. Yes. Okay. Thank you. Thank you. Thank you, everyone.